Hello, everyone. Welcome to today's live broadcast, Exploiting pH as part of your RPLC method development strategy. I am Kristen Moore, multimedia producer of LCGC North America, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LCGC North America and sponsored by MacMod. A company should be able to condense its mission statement into a simple phrase. MacMod's mission statement is smarter chromatography. But you may ask, just what does smarter chromatography mean? It means that when they make a recommendation to you about an HPLC or UHPLC product, it is to partner with you to offer innovative solutions to complex problems. They use advanced state-of-the-art analytical software to match their product portfolio with your separation needs. They leverage their company's 29 years of experience in the LC column business, their technical expertise, and manufacturing network to help you solve your toughest application problems. MacMod provides up-to-date and accurate technical catalogs and reports from industry-leading separation scientists to keep you informed of new technologies. That is what smarter chromatography means. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the red Q&A widget at the bottom of your presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark Help widget in the dock of the bottom of your presentation window. Please note, a copy of today's presentation along with valuable resources is located in the green resource widget on the left-hand side of your screen. I would now like to introduce today's speaker. We are very pleased to be joined today by Dr. Thomas J. Wege. Dr. Wege holds a Bachelor of Science degree in chemistry from Michigan Technological University and a PhD in analytical chemistry from the University of Colorado in Boulder. After obtaining his PhD, Tom joined DuPont Agricultural Products, now DuPont Crop Protection, as an analytical chemist. While with DuPont, he developed a wide variety of GC and HPLC methods to support product development and improvement, registration, and commercialization of a variety of herbicides, fungicides, and insecticides. Starting in 1987, he initiated and championed the use of computer simulation, dry lab software for HPLC separation, development, and optimization. Tom was a leader in the development of procedures and best practices for method development, validation, transfer, and implementation, including guidelines for change management for analytical methodology among R&D, global manufacturing, and registration. After leaving DuPont, Tom provided expert technical support for Agilent Technologies, HPLC columns, and consumables for 9 years, and since 2008 for MacMod Analytical. He enjoys helping customers with pre-sale and post-sale technical support and also develops applications, writes technical reports and marketing literature, and presents posters and presentations for customers and technical venues. Tom, thank you for joining us today. You have the floor.
Well, thank you very much, Kristen, for the very nice introduction and for the nice review of MacMod Analytical's objectives in helping our customers. As Kristen said, the title of today's presentation is Exploiting pH as part of your reverse phase method development strategy. Let's begin. These are the key learning objectives that we presented in the announcement for this webinar today. First, we will revisit the importance of selectivity and resolution in reverse phase separations, and then we will look at which LC parameters affect selectivity most for both isocratic and gradient separations. I will introduce and describe the features and advantages of ACE's new ultra-core solid core columns with their broad pH usability for UHPLC and HPLC separation. Next, we'll look at how to use mobile phase pH and other parameters as part of an effective reverse phase method development strategy. We'll examine some different strategies for method development ranging from relatively simple to very complex for more difficult samples. And at the end of the presentation, toward the end, we will review some data that I generated in preparation for a presentation given at PitCon 2015 earlier in March. Here's an outline of today's presentation. We'll start with a brief review of resolution and selectivity. We'll examine some of the key factors affecting selectivity. I'll show a slide on how the various parameters that affect selectivity are prioritized from work done by Lloyd Snyder, John Dolan, and Peter Carr. We'll look at some method development strategies for both screening and optimization of methods. I'll introduce and describe the ultra-core super C18 and super phenylhexyl phases. Talk a little bit about what PBT is. We'll look at how orthogonal these various ultra-core phases are and in combination with changes in conditions of the method, including solvent, pH, and so on. We'll look at a recommended method development strategy for using ultra-core phases. And then I'll show some real-world examples from my PitCon 2015 talk, including 2 different samples in method development for appetite suppressants as well as for drugs of abuse. I'll briefly show a couple of the new applications on ultra-core phases, in particular one on LCMS. I'll conclude with a summary, and then we'll take your questions, and hopefully I can provide some good answers for you. Let's begin with resolution. Obviously, when you're doing any kind of an analysis, you get your maximum accuracy and precision by having your analytes resolved, but you don't want to have too much resolution, otherwise you're wasting time in your separation, and as we all know, time is money, and we want to get the most data and results to make the best decisions we can, the fastest we can. In general, the resolution value of 1.5 indicates baseline resolution for 2 equal-size peaks, but most people who are doing method development shoot for 1.7 to 2.0 as a minimum, so that there's some leeway there as column ages and you can get more lifetime out of your column. The resolution equation is given on the left side of the slide, where you see that the retention time difference times 2 divided by the peak width sum is the value you get for resolution. So, in looking at resolution itself, it's comprised of 3 different factors, an efficiency factor, a selectivity factor in the middle, and then a retention factor. These come together to define what the resolution is, and these can be more or less changed independently to improve or change your resolution. And here, this slide is meant to show you that over the range of values that these factors take on for most HPLC work, that the selectivity, or alpha, is by far the most powerful of the 3. Admittedly, since 2003, with the introduction of sub-2 micron particles, followed by the introduction of the first 1,000-bar hardware in 2004, subsequently followed by the introduction of sub-3 micron superficially porous particles by Advanced Materials Technology in 2006, we've all become very enamored with using efficiency to affect separations in HPLC and UHPLC. Nonetheless, selectivity is still king and can be used to dramatically change resolution. But how do you know which parameters or factors affect selectivity and which ones are the most powerful? On this slide, I've listed the parameters that affect resolution and selectivity the most for isocratic separations on the left and for gradient separations on the right. The most powerful ones include the stationary phase type, the pH of the mobile phase for ionizable compounds only, the organic modifier type, that is aceternitrile methanol, a blend of the two, etc., percent organic modifier, and then the lesser ones, column temperature, choice of buffer, meaning like phosphate versus acetate, and then the buffer concentration is one of the least ones. You could also throw in here the ion pairing additive concentration if you wanted to for those types of separations. For gradient separations, you have all of the same parameters that you have for isocratic, obviously, plus gradient steepness, or little b, which is equal to proportional to 1 over the K star value. And that K star value, the average gradient retention factor, is given by the equation here in the middle of the right-hand side of the slide. And it's a function of gradient time, flow rate, the difference in the organic modifier concentration, the column volume, and then a factor that's a function of the molecular weight of the compound. And it's really obtained from a plot of log K versus percent organic for a given analyte. So you can see that the column volume, which is embedded in the K star equation, is really a function of the column length, its internal diameter, and the porosity of the phase that's packed inside. Another factor that's often overlooked by people doing method development and method transfer is that the dwell time, which is also borne out by the ratio of the gradient volume to the actual column volume, that must be kept constant. Otherwise, you will see some minor changes, particularly in the front half of a separation when you move from one instrument to another, as well as from one column size to another. Sorry, the slides are changing a little slowly here. On this slide, I'm showing some factors prioritized based on a publication by Lloyd Snyder, John Dolan, and Peter Carr. And by far, the pH of the mobile phase is the most powerful factor for changing selectivity, followed closely by organic modifier, gradient time, the phase of column that you're using. The percent organic modifier and column temperature are pretty much equal, and then followed last by the buffer concentration. And in this paper that I've forgotten to put the reference for here, but I can make that available to you by email or otherwise, what they've described there was that for equal size peaks with a resolution of 1.5 for a common size of column that's often used, which at that time was a 4.6 by 150 5-micron column. Here I'm showing a 2.1 by 50 millimeter UHPLC column, either a superficially porous solid core or a sub-2 micron column. And with a K value of 1, you come up with a log alpha value for separating two co-eluting peaks of 0.05, and Snyder proposed doubling that to 0.10. So that gives you an idea in a mental calibration of how impactful these various factors are. What's interesting also is that if you change more than one at a time, what you actually get for the delta log alpha value in the third column of the table is the square root of the sum of the squares. So if you take, say, for example, organic modifier and gradient time and change those simultaneously, the factor then becomes, the delta log alpha value becomes the square root of 0.2 squared plus 0.2 squared. So it magnifies the impact when you change more than one factor at a time. Just a quick comment about the importance of pH for ionizable compounds. Traditional wisdom and recommendations from the experts in the field have said that you really should begin your method development work at low pH, because that's the pH for where silica-based columns, all of the silanols that are unbonded or un-end-capped, are fully protonated, and your bases are positively charged but will tend to interact least with your silanols. Your neutrals are unaffected, and your your acidic analytes are fully protonated or mostly protonated and are going to be best retained. However, there's a lot of times where type B phases may not perform so well for bases due to overloading, due to being too inert or being too free of metals, etc., and free of silanols, that they actually will tend to give poorer peak shape at low pH. So it's beneficial to be able to access more of the pH range for both bases and neutrals and acids. And I, for one, have not had a lot of experience over my years of working in LC with the higher pH range, simply because there weren't a lot of good columns around that could handle mid to high pH. Now I believe we have some that can do that in the ultra-core phases. This next slide shows an example of different method development strategies of varying complexity for doing systematic method development, starting with the least complex samples and going to the right with the most complex samples. And these are scenarios in which you would say in the first box on the left top row there, one column phase, one temperature, one pH, possibly two organic modifiers, meaning aceternitrile and methanol, for example, and one gradient time, which you might pick to be an effective K-star value of like 3 or 5 or something like that. Set your flow rate for ideal for your particle size of interest. You try aceternitrile and methanol separately with your given pH that you've selected, and that may be adequate to develop a separation after you optimize your gradient or move to the appropriate isocratic conditions. However, as your sample complexity increases, it's prudent and wise to consider either one phase that does well in terms of peak shape for your analytes and one temperature, and maybe you try two pHs, one or two organic modifiers, etc. In the far right box that I've highlighted here, you might consider using two or more phases, maybe one temperature, 2 or 3 different pHs or more, 2 organic modifiers, and you can really take your sample and profile it under a variety of conditions and then choose which condition you find to be the best in terms of peak shape, retention, best chance to optimize the separation further, etc. In the lower line of boxes and scenarios for under optimization, we see that I've highlighted one there in terms of taking that a step further. Once you've found a condition that looks promising, then you can approach it by doing ideally some computer simulation in which you change the variables in a systematic way and then can use computer modeling to find the best place to work that's most robust and gives you the desired resolution for your poorest separated peak pair. Now I'd like to take a few minutes to introduce the ACE ultra-core solid core columns. They're available for UHPLC/HPLC in a 2.5 micron particle size, as well as a 5 micron particle size for purely HPLC separations, and by that I mean 400 bar or below. The 2.5 micron particle can be used with HPLC systems, UHPLC systems, and UPLC systems. It's available. The ultra-core are available in super C18 and super phenylhexyl, and these are two selectivities one of which offers hydrophobic only interactions, and the other offers both hydrophobic and pi-pi interactions. The hardware that these are packed in is stable up to 1,000 bar, which is about 14,500 psi. And they do use 2 micron inlet fritz, which helps give you longer uptime and column life, as well as improved ruggedness. And these phases are at low pH, of course, good for many column volumes as you can deliver ordinarily like you would get from any other column material, but they are targeted for 20,000 column volume lifetime minimum at 40 degrees or below at the higher pHs, 8 to 11.0. So what gives this material, these two phases, their unique performance? It is encapsulated bonding technology, or EBT. This is just a diagram that shows a traditional C18 bonding for a material on the left-hand side of the graphic at the top, and where you see your unbonded or residual silanols. You see some silanols that have been end-capped with an appropriate end-capping reagent, and then you have your two C18 chains. On the right is a cartoon of what the super C18 and super phenyl hexyl would look like. In this case, the super C18. You have actually a inert layer that is on the surface. It is not a polymer per se, but it's a proprietary end capping that gives improved and superb peak shape for bases, acids, and neutrals over a broad pH range. And it gives dramatically higher ligand coverage and effectively eliminates silanol interaction. That excellent coverage also gives the column the stability at mid and high pH, but you'll get your maximum lifetime with volatile buffers at higher pHs, which means in general, it's been shown back in the 90s that mobile phases such as phosphate and carbonate, when they're mixed with organic solvents like acetylnitrile methanol, tend to have a higher apparent pH than they would if you're using something like ammonium acetate with or without ammonium hydroxide adjusted to higher pHs. So if you stay with those things, I don't believe I have any data to show on other organic type buffers, but I would imagine that those will also give extended lifetime. The other really nice feature of these packing materials is that you don't see any memory effects or hysteresis from switching between eluents at different pHs. So you can move from low pH to high pH, back to low pH, back to high pH, and the columns still perform nicely and consistently. Now, just a brief introduction to the way that orthogonality of phases and conditions are often measured by many now. The late Uwe Neue of Waters came up with a selectivity descriptor S that is a measure of orthogonality, and it's given by the equation there in the top left of this slide. And example plot, scatter plot is shown on the top right. And what he did was take the coefficient of determination from a scatter plot of retention times from analytes obtained under gradient conditions for two different columns or the same column under two different conditions. You plot one versus the other, and you look at the scatter, and you take the R-squared value and put it into the equation there, and you end up with a number that has some meaning in terms of orthogonality. The smaller the number, the less orthogonal. The higher the number, the more orthogonal or different in selectivity. And of course, that's only a value if you have a significantly large set of analytes to make those kind of measurements with. Or you also have to have a diverse set of analytes as well as a larger sample set. If you only had 5 analytes, it wouldn't mean a whole lot. But in this case here, on the bottom right, we see the results from principal component analysis of the 50 analytes that they used for this characterization of these columns, and it's quite a wide spread of selectivity involving analytes such as acids, bases, neutrals, and some other ones included in that set. So it's a quite wide distribution of analytes there that can be used to mean something for future prediction. So couple quick examples of what these plots look like. When you have low selectivity differences between columns under the same conditions or same column under different conditions, you see a plot much like you see here in the center of this slide, a very low S value of 3. This is something that you would observe when you plot the results from two different vendors or with the same phase or the same retention mechanism, same solvent, under the same conditions, or if you were to say compare a C8 phase versus a C18 phase from the same manufacturer, you're going to get a very high R squared number, very little scatter, and a very small S number. If you look at something that might be representative of moderate selectivity differences, say between maybe slightly below 10 to below 30, these are quite moderate and important and useful selectivity differences. This plot shows a selectivity value of S value of 22 for same column ultra-coarse superphenyl hexyl analyzed with this sample set with acetonitrile in one case versus methanol, and these were all carried out at high pH. And this is the type of scatter plot you would see, and you could say, well, that doesn't seem like it's very scattered, but it significantly is. And even a number as small as 10 or between 8 and 12 is a useful enough difference in selectivity that you can move peaks around. And we saw that with some of the work that ACT has done with some of the other totally porous phases that they have introduced, including like the C18 PFP and the C18 AR phases. This is an example of very high selectivity value differences, an S value here of 81, and quite a broad scatter in the scatter plot here. This is for ACE ultra-core super C18 separations of these 50 analytes in acetonitrile and comparing low pH in the y-axis versus high pH in the x-axis. This is the kind of plot that you would expect to see when you're changing multiple variables, something greater than 30, not necessarily 81. Again, when you are changing pH, you want to do that with a column that can handle the other conditions. Obviously, you don't want to put it, carry out method development on a column that's going to last you for a few days or a week. Here's an example of a separation carried out at low pH with methanol in both cases for the super C18 and the superphenyl hexyl. And here you have an S value of 19 for a fairly large set of analytes, up to about 23 components. And highlighted by the circles are some of the differences in the separation between the top and the bottom. Another example, this one with superphenyl hexyl in both chromatograms between low pH with methanol and high pH with methanol, the upper trace being the low pH and the bottom trace being the high pH. And this scatter plot is really like a shotgun pattern or whatever. It's is consistent with a selectivity S value of 83. And here is a case with a mixture of acids, bases, and neutrals. Case here, 11 analytes, and peaks are moving around quite considerably. This next slide shows experimental design combined with the S values for the various combinations for low pH, namely pH 3 in the bottom left-hand side of the slide versus pH 10.7 with ammonium hydroxide for the two phases. In these cases here, the corners of this square, red ones are the ones for acetonitrile, and the blue circles and designations are for methanol. And then the numbers that are along the diagonals and along the sides of the square represent the S values that they obtained for, for comparison of those 50 targeted analytes that they use for the comparison of orthogonality. The average orthogonality value, or S value, is 24 between acetonitrile and methanol, and between the two phases, 13. Again, I mentioned that even a number that size is still adequate to move peaks around for method development purposes. Looking at it in another plane, you have in the upper plane the red circles, or filled in circles. You have the two different phases in acetonitrile. In the bottom plane, trapezoid, you have methanol in the blue circles, and you can see the big changes for both phases, moving from low pH to high pH in either acetonitrile or in methanol. Here, the average S values switching between low and high pH is 82, and between the super C18 and the super phenylhexyl, it's 13. Similar results here, switching between low and high pH with methanol and acetonitrile, and then either the super C18 or the super phenylhexyl. Looking at it a different way, you see the numbers averaging around mid-20s for between the two phases, and 82 between low and high pH. Again, the red circles represent acetonitrile mobile phases with the two stationary phases, and the blue dots represent methanol for the super C18 and the super phenylhexyl. Putting that all together, you have a diagram, and if you were to switch from, say, acetonitrile in the upper left of this rectangular prism down to the bottom right with methanol in the rectangular prism, that diagonal of that prism is S value of 85. So when you change mobile phase, pH, stationary phase, and solvent, you get, again, a very large change in selectivity, a very large orthogonality. So to kind of capture all of that in a method development strategy, you might consider the following as shown on this slide here. You can use these two phases with two temperatures at 1 pH with two organic modifiers, and then maybe use a couple of different gradient steepnesses to provide the data that's necessary to provide you with both screening in combination with at least a partial optimization of the separation, as shown by the leftmost square in blue. The middle one, you work with one column at two different temperatures, 1 pH, two organic modifiers, and two gradient times. Again, another way to approach the same or a different problem. And then on the far right, you use two or more different column phases, two temperatures, two pH values, and two organic modifiers with two gradient times. Usually you need 3 to have a checkpoint in the middle. And here, example is pH 3 and pH 10.7. And that would provide enough data for input into a software package like DryLab and do a actual 3D cube optimization where you can actually find the best place to operate within an entire cube of an experimental design in a working space. So that's an example of how you can use the various columns, mobile phases, pHs to really tackle very difficult separation and method development problems. Now, what I'd like to do is switch gears just a little bit and kind of show some of the findings that I had with my first experiences with working with these phases in preparation for a presentation earlier this month. And my objective really was to explore the performance of these columns, and I was starting out by working with a couple of fairly short, narrow diameter columns, 2.1 by 50 millimeter long, super C18 and super phenylhexyl. I wanted to examine how they performed in both acetonitrile and methanol, and I wanted to take a look really across the pH range, starting with 2.8, 3.8 at low pH, and then some of the higher pHs surrounding the pKa for ammonia, which is at 9.2, so plus or minus 1 pH around the ammonia pKa. And then also, I'm not in data not shown here, but I did some work at intermediate pHs surrounding the 10.2 pH as well. For this work, I worked with a Shimadzu 600 bar system. I believe it has a delay volume of about 0.4 milliliters, 600 bar max. And the samples I chose were a mixture of appetite suppressants for the first sample type, and then a mixture of 16 drugs of abuse for the second sample. Here's the first sample, and it consists of these 9 compounds, methamphetamine through phenylpropanolamine. Again, I worked with the columns as I described in the previous. I was working primarily with ammonium formate at lower pH, 2.7, or say 2.8 and 3.8. From earlier work, I found that it really, for a lot of these mixtures of bases, that it's primarily productive to work around the pKa's, say in the 2.8 to 3.8, maybe a little bit higher for bases when bases are involved in the sample. And also, it's very useful to work at higher pHs in the 8.2 to 10.7 range. For gradients, for screening, I wanted to work between a fairly low organic and fairly high organic to make sure that I was seeing all the peaks, because I'd never worked with these samples before directly. And as you'll see in some of the slides, I originally started out with working using a gradient from as low as 2% organic going up to 30%, which I quickly found was not adequate except under the very low pH conditions. What I found was that, of course, as bases become more neutral in their character as you move to higher pH, they're retained more, and you need to be able to go up to higher percent organic in order to elute them off, and that's particularly true for methanol versus acetylnitrile, as methanol is quite a bit weaker. These are the structures of the compounds, and you can't really see them as well as I could on my PowerPoint slide, but nonetheless, they're shown here, and you'll be able to see those in the PDF. In the table on the right, I have listed the pKa's, the log P's, and the PI's for the various compounds if they have them, as well as the last column, which is the retention time with acetylnitrile and the pH 10.2 buffer with ammonium acetate adjusted with ammonium hydroxide on the 2.1 by 50 and so on, with the conditions shown there, and they're listed in increasing elution order or retention time. All of the log P, PI, and pKa values were taken off of chemicalize.org, which will do a nice job of presenting calculated values for those for use for deciding things and doing your method development work. The reason why I chose the appetite suppressants was that I had a customer that called in middle of last year from the FDA, Phyllis Wilson, and she was charged with developing a LCMS method for a variety of these appetite suppressants. I think there were 14 or 15 compounds in her sample. And she had tried a lot of method development work from low pH to mid pH, and actually had found that pH 5.1 was the best for her sample up to that point. And she had tried, I think, some different C18 phases as well as a phenyl phase from some competitors, and gave us a call. And I thought it would be a good opportunity to work with her, not only to see how some of our totally porous phases worked, but also to see how well the totally porous super C18 phase worked. We also have, just as an aside, we do have ACE Excel super C18 columns in hardware that goes to a 1,000 bar, and it has the same performance that the UltraCore super C18 has, but is in a totally porous particle size ranging from 2 to 3 to 5 for HPLC and UHPLC application. But I'm showing this just because of wanted to show, I recommended her that she purchase a C18 PFP, and she tried it at the same conditions that were her best up to that point. And as you can see, some of the peaks for bases don't do so well either at very low pH or at mid pH with any kind of a ultra pure, low metal content, well end-capped phase. See the peak shapes. This is well known and documented by David McCauley and others. And the lower trace, so after that, and some method development work with different gradient steepnesses didn't help much. And then when you have bad peak shape, you pretty much either have to live with it or you want to try to find something else. So I sent her a column that I had, a Super C18 in the same dimension, in the 2-micron, totally porous. And it gave somewhat improved, in some cases, peak shapes and so on for this sample set, but still wasn't really where she or I wanted it. So she agreed to do some additional work. She had never worked at higher pHs, so we pursued that. We tried separations at pH 8.2, 8.7, 9.2, 9.7, 10.2, 10.7, so on, just to kind of do what I did with the samples, as I'm going to get to here in a second. And as it turns out, you can take dry lab software, and if you take even a gradient separation and kind of like using dry lab in a little bit different way, you're actually putting gradient data into the pH optimization and retention times, but it will produce a window diagram, and it predicted that pH 9.3 was the optimum, and it did a pretty nice job of predicting how things would look. And this is like I said, it's ammonium acetylnitrile gradient from 10 to 90%, a fairly simple thing. We didn't even go beyond this in terms of optimizing a gradient steepness or any kind of a gradient program. But it just basically just dropped it into the same gradient that she had been running, but at that desired pH. And I thought it came out quite well in terms of the peak shapes for all of them, and then improved resolution and selectivity. The one thing you have to be careful when you move to other pHs is that, of course, the UV absorbance can change for analytes as you move from one pH to another, especially dramatically changing pH. But also, your mass spec sensitivity can also change. So you have to everything in chromatography and in life is a compromise. So you have to dial all those things together in terms of what your objective is versus what you want. You can get pretty peaks, but if you can't see them, it doesn't matter. So anyhow, I just wanted to kind of go through that first just to show you what that. So I chose a few of the compounds from her sample set, which I described in earlier slides. And so I thought I would do some experimentation here. I'm showing on this slide two different pHs, low pH at pH 3.8 with ammonium formate and higher pH at 10.2 with ammonium acetate adjusted with ammonium hydroxide. The traces on the left ran from 2 to 50% organic for both acetylnitrile on the top and methanol on the bottom left. And similarly, the acetylnitrile is on the top on the right, and on the bottom right, it's methanol. You can see that I had to go up to 70% organic for the two traces on the right-hand side of the slide because the retention increases quite a bit when you raise the pH for all these. These are all basic analytes. So I've circled the peaks that were poorly separated there, in particular at the early part of the run, and there was another pair just before the large chlorodiazepoxide peak there in the upper right trace, for example, that was partially resolved under the conditions of this. When I did a lot of this work, I tried to fix the K star value at about 14, and it's an odd number, 14.3. These were all carried out at 0.6 mils a minute at 30 degrees C. And again, these are screening gradients meant to kind of show, how do the different conditions perform in the different phases. So that's what I learned from this simple step here of running these under these various conditions. Nonetheless, you can do optimization on one of those conditions that you most like the most, or in this case here, I'm showing some examples of two gradient runs from 2 to 70% in 10 minutes in the upper left trace and in 20 minutes in the bottom left trace. If you input those data into DryLab, as simple as those chromatograms are, you can come up with an optimized separation. Shown on the right is the 3 by 100 millimeter column, 2.5 micron, 1 milliliter per minute, and it's a linear gradient from 15 to 60% in about 10.5 minutes at pH 10.2. Again, at 30 degrees C. So, it came out nice for that. It wasn't as complex of a sample as what she had, but it was a way of coming up with a separation like that, and also showing how you can scale up. Again, the linear velocity for the left-hand chromatograms is not the same. Take that back. This one, we are using the 3 by 100 in both cases. You'll see in some subsequent slide that worked from the 2.1 by 50 to the 3 by 100. The next sample I dealt with was a sample of 16 drugs of abuse, and those include normorphine, morphine, and so on, as shown in the table on the right-hand side of the slide. I've highlighted the compounds that are isobaric, that have the same molecular weight, in the same color, so you see that normorphine and dextromethorphan are very comparable in molecular weight, but in terms of the retention time, in this case on the superphenylhexyl in a low pH gradient from 2 to 50% acetonitrile, they're at the opposite ends of the chromatogram. And similarly for the other colored isobaric compounds, they're well separated under the pH 2.8 conditions, and they also are nicely separated on the optimized conditions you'll see in a few minutes. This slide compares the UltraCore Super C18 2.1 by 50 size with 4 different pHs using acetonitrile. The upper left is 3.8 pH, the upper right is 8.2, the bottom left is 9.2, and the bottom right is pH 10.2. Again, at the higher pHs, it was necessary to use a higher top end for the gradient in terms of percent organic, and I did attempt to keep the K-star value the same at 14.3. That can easily be done with Excel or other means. Do that. So you can take a look at these and you can say, where is the best place to operate, or which of these conditions looks most promising in terms of peak shape and in terms of potential for further optimization. And personally, I'm looking at these. The upper left looks to be a good candidate with a pH 3.8, and really the bottom right also turned out to be, but even the 9.2 pH could be easily used for that. In this case, I compared the superphenylhexyl phase in the same size at 4 different pHs. They're the same ones as I described in the previous slide, 3.8 top left, so on, 8.2 in top right. Here you can see that the 8.2 and 9.2 give some misshapen peaks, and you're going to see some kind of strange behavior sometimes for certain analytes, and it's very analyte dependent. So really the top left and the bottom right at the 2 pH extremes turned out to be the ones that have the most potential for additional optimization. This is a slide showing 15-minute and 30-minute gradients left and right side of the slide. Separations carried out on a 3 by 100 size, 2.5 micron for the 16 drugs of abuse on a superphenylhexyl UltraCore, and you have nicely all of the components separated, although there's one peak pair that's partially resolved there, as shown in the 2 blown up regions of the chromatograms. This slide shows the translation of the gradients from a 2.1 by 50 UltraCore superphenylhexyl with methanol in the top trace, carried out at pH 3.8 with ammonium formate, and then moving to a 1 millimeter separation on a 3 by 100, going from 2 to 70%, and all of the peaks are nicely separated there. Bottom left is the comparable separation for the same sample on a 2.1 by 50. Again, the K-star is 14.3, scaling that up to a 3 by 100 in a 2 to 95% gradient in both cases. The translated gradient shows nicely a nice separation of all these components. I'll close with a couple of examples here from a number of new applications that have been done with collaborators or done in the laboratories at ACT. This shows the power of what you would ordinarily think is a pretty weak in terms of resolving power size column, a 4.6 by 150, but this has 2.5 micron particles, and columns like this size in a 150 length in this particle and in this solid core material can deliver efficiencies ranging from probably 30,000 plates to 40 plus thousand plates, and really work well even as a single column in an impurity method. What's nice is, and that can be used on an HPLC or a UHPLC system. If you're constrained to an HPLC system, you can use a 5 micron column in this solid core, superficially porous material, and you can concatenate 2 columns together in tandem. And if time is not the issue, you can still operate at HPLC pressures and get really fantastic resolving power. Those columns probably run around 28 to 30,000 plates each, so you've got maybe 55 to 60 plus thousand plates with 2 columns in tandem for a 5 micron. A brief LCMS example. This was submitted by a collaborator to Dr. Alan McEwen at Advanced Chromatography Technologies. These are tetracarboxylic acids, which are very hydrophobic. There are 80 to 82 carbons in these analytes, and this is just an example that shows how very unusual and very difficult samples can be handled with these products, and they really are conducive to LCMS applications. In fact, one of our subsequent webinar webcasts will be given by Alan later in this year. Finally, to summarize the objectives that we had are up on the top of the slide, and basically we've revisited the importance of selectivity on resolution and talked about the factors that affect selectivity the most based on actual research done by Snyder et al. And we talked about how they could be used in your method development strategy. I described the new ACE ultra-core solid core columns for UHPLC and HPLC and showed how they could be used over a broad pH range to provide very powerful tools for method development. Showed you diagrams of how they were orthogonal in selectivity when you combine phase change in concert with pH and organic modifier changes. And we also talked about and discussed some method development strategies for simple samples as well as for very challenging samples, how you can use the combination of various organic modifiers, stationary phases, and mobile phase pH changes to affect even very difficult separations with 10, 20 or more analytes. And with that, I would like to thank you for attending today, and I would like to remind you that MacMod is a relatively long-time player in the HPLC and UHPLC market, been around for almost 30 years, and we are representing ACE, Halo, and Protosil columns for quite a long time, and we're the exclusive distributor for ACE and Halo and one of the main U.S. distributors for Protosil columns. And with that, I am open for questions.
Thank you, Tom, for that informative presentation. Before we get started, we're going to take 2 brief polling questions. You can click directly on your screen to submit the question. Let's pull up the first question. Would you consider using pH as a selectivity modifier after attending this webinar? Again, you can just click directly on your screen, and our choices are yes or no. Okay, great, thank you. Let's move on to the next polling question, please. Would you consider using mobile phase pH between 8 and 11 after having attended this webinar? Our choices are yes or no. Let me repeat the question. Would you consider using mobile phase pH between 8 and 11 after having attended this webinar? Thank you for taking those two brief polling questions. We're going to move right into the question and answer portion. As a reminder, you can submit questions by typing them in the Q&A box, which can be found by clicking on the red Q&A widget at the bottom of the presentation window. Okay, Tom, our first question is, can you please explain once again what is the orthogonal column? I know about orthogonal selectivity like Hillock in some applications, but not columns. Thank you.
Well, certainly different modes of chromatography can be orthogonal, like Hillock compared to reverse phase separations. But in the reverse phase mode, there are columns that are very similar in selectivity and retention, or similar in selectivity but not so much in retention. But there's also a number of different tools that are out there right now that go beyond what some of our customers have been using for a long time, which is, percent carbon load and, porosity and bonded phase load in terms of or surface area, meters squared per gram, and so on. There are tools out there like www.hplccolumns.org, and the USP site also has the same database that was really developed from work done over a long period of time between Snyder, Dolan, and Carr and collaborators. Now that work is being carried on after John Dolan passed the torch to Dwight Stoll at the university in Minnesota, and he is the one that is continuing on with characterization of columns, and then puts the data up on the web and on the USP site so that people can make scientifically based empirical decisions about which columns are closest and which columns are most dissimilar in selectivity.
Excellent, thank you. And this question kind of goes hand in hand. Can you please explain a little more on using two orthogonal columns in tandem?
I'm sorry if I gave that impression. Actually, that is a technique that is being considered more about putting two different phases in tandem directly to give you kind of like a composite phase, if you will, or composite column. But what I meant was putting two columns of the same stationary phase in tandem to give you twice as many plates, and of course the square root of 2 times the number of the improvement in the resolution. So now using two columns with different selectivity in tandem, that probably goes beyond the scope of this seminar or webinar, but I'd be happy to answer that one afterward. But that was not my intention in mentioning that. Those were the same phase in the same geometry, or maybe even a shorter geometry, but ideally to multiply the amount of resolving power that you have.
Great, thank you, Tom. Our next question is, have you seen any instrument compatibility issues at the very high pH near 11?
The highest I've worked on a consistent basis was 10.2, although I did do some work at 10.7. Some instrument manufacturers, if you're going to work exclusively at high pH for a long period of time, I know Agilent, having worked there, they have a high pH kit that you can modify your various models of your HPLC system with to get rid of some of the Vespel and so on, and replace the polymers with other ones that can handle the high pH better. But I've worked at high and low pH over a month's time here and there, and have had no observed problems with any damage to the instrument. There has been some talk in the past that operating at very high pH, maybe it's worse with phosphate and carbonate buffers, but that you can start to slightly cloud your quartz flow cells, but I have not seen that, attempted to measure that.
Thank you, Tom. And we have time for one final question today. Is there a guide, flowchart, or program that can calculate best type of column and eluents with MS modifiers based on the analyte, chemical properties, and matrix for LC-MS? What is the precision of this type of software?
What is the precision?
Yes.
I don't believe there's anything out there that can calculate the best combination of column and eluent. A lot of this is, done basically by empirical measurements. You need to find out first, where your analytes will ionize best if you're talking about MS modifiers. And then once you know where they ionize best, since you want to detect them the best, especially if you're challenged with impurity analyses or very low levels to measure, then once you have that information, then you would use those conditions and probably those conditions only to proceed with evaluating in a systematic and empirical way which is the best place to work and what knobs you can turn within those constraints relative to your signal. But I'm not aware of any guide or flow chart or whatever, but I'd be happy to discuss that with the person afterward or by email or otherwise.
Great. Thank you, Tom, and thank you for that excellent presentation today. Unfortunately, we are out of time. I would like to thank the audience for their interesting questions and participation in today's event. I would also like to thank our sponsor, MacMod, for making today's educational webcast possible. If we did not get to your question today, we will follow up via email after today's event. Today's webcast will be available for on-demand viewing through March of next year. You will receive an email from LCGC alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.